Hello everyone, thank you for tuning in. We'll be talking about DNA replication.
The first concept we're going to explain is base pairing. Basically, there are four types of nucleotides and their names are adenine, thymine, guanine, and cytosine. Adenine hydrogen binds with thymine and guanine with cytosine. The parent strand of the DNA serves as a template for the new strand, which has nucleotides complementary to those of the parent strand.
In the beginning of the 1900s, scientists were trying to figure out which model would suit replication. There were three different types of hypotheses, including the conservative model, the semi-conservative model, and the dispersive model. The conservative model said that two new daughter strands would combine to form one molecule of DNA, and the two parent strands would join to form a separate molecule. The semi-conservative model stated that in each double helix there would be one new strand and one old strand. The dispersive model said that each molecule was made of a mixture of both the new and old strands.
In the 1950s, two scientists, Messelson and Stahl, designed an experiment to determine which model was correct. In their experiment, cultured bacteria were immersed in nitrogen-15 medium, then transferred to a nitrogen-14 medium. The first generation of replication, which was centrifuged after 20 minutes, contained a hybrid of the heavier nitrogen-15 and the lighter nitrogen-14. The second generation of replication, which was centrifuged after 40 minutes, contained both the hybrid and the pure nitrogen-14. The results after 20 minutes thus had a single band in the middle of the test tube, indicating the hybrid DNA. This eliminated the conservative model, which predicted two separate bands. The results after 40 minutes contained the hybrid band and a separate band that was less dense. This ruled out the dispersive model, which predicted a single hybrid band. In conclusion, DNA replication
Replication begins at segments of DNA called the origin of replication. In organisms with singular circular chromosomes, there are only one origin of replication, whereas in organisms with linear chromosomes, there are many origins of replication. As DNA is replicated, replication levels expand from one origin of replication. At the end of each replication level, there are two replication forks. In organisms with multiple origins of replication, the bubbles eventually unite.
There are many proteins involved in DNA replication. Helicase unwinds the parental DNA at the replication forks. Notice it is a wrench. Single-strand binding proteins stabilize the single-stranded parental DNA by binding to it. Notice it is a stapler. Topoisomerase reduces the stress ahead of replication forks by breaking and rejoining DNA strands. Notice it is an icy hot sleeve. Primase synthesizes an RNA primer at the 5' end of each strand. Notice it is primer. DNA polymerase III synthesizes new DNA using the parental template and adding nucleotides to the 3' end of the primer or existing DNA strand. Notice it is Mario with a hammer. DNA polymerase I replaces the RNA nucleotides of the primers with DNA nucleotides at the 5' end. Notice it is a stapler remover. DNA ligase joins the beginning and end of two DNA segments. Notice it is Elmer's glue.
In our diagram, replication is initiated when helicase unwinds the parental DNA, topoisomerase relieves the stress of DNA after the replication fork, single-strand binding protein attaches to the strands to keep them separated, and primase synthesizes the RNA primer.
As the new strand grows, nucleotides, which come from nucleoside triphosphates, are added. The nucleoside triphosphates have a lot of chemical energy because their triphosphate tails all have negative charges. As each nucleotide is added, two phosphate groups are lost as a molecule of pyrophosphate. Next, the pyrophosphate is hydrolyzed to form two molecules of inorganic phosphate. This hydrolysis is exergonic and coupled with the polymerization of DNA.
In our diagram, DNA polymerase III adds nucleotides to the daughter DNA molecule after the primer. DNA polymerase I removes the RNA primer and replaces it with DNA nucleotides. The resulting segments are joined by DNA ligase.
The behavior of replication is influenced by DNA polymerase III's inability to add to the 5' end. Because of this, there are leading and lagging strands in the replication bubbles. In the leading strand, nucleotides are added continuously from the 5' to 3' end. In the lagging strand, nucleotides are added discontinuously in Okazaki fragments. Each fragment has its own primer, which is acted on by DNA polymerase I and ligase.
This is our interactive model.
So now we're going to make an interactive model using all of the enzymes involved in DNA replication. And here's our bank of enzymes. And remember that every process in biology is based on the interaction of different proteins. So first we're going to take helicase, and we're going to take it over to our double-stranded DNA molecule here. And helicase is going to unwind it to make a replication bubble, just like that. Thank you, helicase. And next, the single-stranded binding proteins will bind to each of these single strands to keep them from binding back into the double-stranded DNA. And topoisomerase will enter into the picture to make sure that there's not too much stress on either side of the replication forks here. So it'll be one here and one here. Next, primase will start a primer. It'll synthesize a primer of RNA monomers. And DNA polymerase III will then be able to add DNA nucleotides from the 5' to 3' end after the primer. And after all these nucleotides are added, this primer, which is composed of RNA, will be replaced by DNA nucleotides thanks to DNA polymerase I. And these two segments of DNA nucleotides will be joined by ligase. So now we'll have a continuous strand of DNA nucleotides.
This next video is in real time.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand.
As DNA polymerases add nucleotides to the daughter strand, other DNA polymerases proofread each nucleotide to ensure that it is complementary to the parent strand. One type of DNA repair is mismatch repair, in which enzymes remove incorrectly paired nucleotides and replaces them with a proper nucleotide. Another repair system, called the nucleotide excision repair, is involved with cutting out whole segments of daughter DNA using the enzyme nuclease, and replacing them using DNA polymerase and ligase.
As we have mentioned before, nucleotides cannot be added to the 5' end. This becomes a problem because the beginning primer cannot be replaced with DNA when there is no 3' end available. After each replication, the DNA is shortened. To prevent the shortening from affecting genes, there is a non-coding segment of DNA called the telomere. The telomere is naturally shortened by an enzyme, but an enzyme called telomerase reduces the natural effect.